This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Welcome. Ever wondered if animals could photosynthesize like plants? This exploration delves into that very question, explaining this complex topic in easy-to-understand language. We'll uncover surprising examples of animals that borrow or steal sunlight's energy, exploring symbiotic relationships with algae, the fascinating phenomenon of kleptoplasty, and even insects that generate electricity from light. Prepare to be amazed by nature's ingenuity and discover how these adaptations could revolutionize food and medicine. Let's dive in. Chapter 1. Stealing Sunlight, the Sea Slug's Secret to Survival. Picture a vibrant green leaf, delicately veined and undulating gently in the water. Now, imagine that leaf is actually an animal, a sea slug named Elysia chlorotica. This remarkable creature, found along the eastern coasts of the United States and Canada, has mastered a survival strategy unlike any other in the animal kingdom. It steals sunlight. Elysia chlorotica, also known as the eastern emerald elysia, isn't your typical garden slug. Its body, a striking emerald green punctuated by small red or white spots, is flattened and elongated, perfectly mimicking the shape of a leaf. This leaf-like appearance isn't just for camouflage. It's a crucial part of the slug's unique ability to harness the power of the sun. The slug's translucent skin allows sunlight to penetrate its tissues, reaching the very organelles that hold the key to its survival. This sea slug's primary food source is a specific type of algae called Vaucheria litorea, a stringy, yellow-green algae found in salt marshes. But Elysia chlorotica's relationship with this algae goes far beyond a simple predator-prey dynamic. When the sea slug consumes Vaucheria litorea, something extraordinary happens. Instead of completely digesting the algae, Elysia chlorotica extracts the chloroplasts, the tiny powerhouses within the algae cells responsible for photosynthesis, and incorporates them into its own cells. The stolen chloroplasts, now residing within the sea slug's digestive tract, don't just sit idle. They continue to perform their primary function, photosynthesis. These kleptoplasts, bathed in sunlight filtering through the slug's translucent skin, absorb light energy and use it to convert carbon dioxide and water into sugars, just as they would within the algae. This remarkable adaptation allows Elysia chlorotica to survive for an astonishing 9 months without eating, relying solely on the energy produced by these captured solar panels. This duration makes it the longest-lived symbiotic relationship of its kind known to science. Unlike most animals, which break down their food into tiny molecules before absorbing them, Elysia chlorotica has specialized cells lining its gut that can engulf whole cell parts, including chloroplasts. These cells form protective pouches around the chloroplasts, shielding them from digestion and allowing them to remain functional within the sea slug's body. The type of chlorophyll produced by these captured chloroplasts is chlorophyll A, the same primary pigment responsible for capturing light energy in plants and algae. This chlorophyll A gives Elysia chlorotica its vibrant green hue, further enhancing its leaf-like disguise and maximizing its light absorption capabilities. The exact mechanisms by which Elysia chlorotica maintains these stolen chloroplasts for such extended periods remain a subject of ongoing scientific debate. Some researchers believe the slugs have acquired algal genes through horizontal gene transfer, allowing them to produce proteins essential for chloroplast function. Others suggest the slugs may have evolved unique mechanisms to protect the chloroplasts from degradation and prolong their lifespan. There's also debate on whether the slugs directly absorb the energy produced by the chloroplasts or simply digest them slowly, benefiting from the stored sugars. Regardless of the precise mechanisms, the fact remains: Elysia chlorotica has evolved a remarkable adaptation that blurs the lines between plant and animal. Its leaf-like shape, translucent skin, and simplified digestive system all contribute to its ability to steal sunlight and thrive on its energy, a testament to the ingenuity and adaptability of life on Earth. This tiny sea slug, with its remarkable secret, offers a glimpse into the incredible diversity of life and the surprising ways in which organisms can adapt to their environment. Chapter 2. Symbiosis. A dance of light and life in the animal kingdom. Photosynthesis, the engine of life on Earth, isn't always a solo performance. In the underwater realm, a fascinating ballet unfolds, a symbiotic dance between animals and algae, where sunlight becomes the currency of life. This partnership, honed over millennia, showcases the remarkable adaptability of life and the intricate web of connections that sustain our planet's ecosystems. Coral reefs, vibrant underwater cities teeming with life, are perhaps the most iconic example of this symbiotic dance. These complex structures, built by tiny coral polyps, owe their existence to a remarkable partnership with microscopic algae known as zooxanthellae. These single-celled algae, primarily dinoflagellates of the genus Symbiodinium, reside within the coral polyp's tissues, creating a mutually beneficial exchange of resources. The algae, safely nestled within the coral's protective embrace, capture sunlight and convert it into energy-rich sugars through photosynthesis. These sugars, the very fuel of life, are then shared with the coral host, providing the energy needed for growth, reproduction, and the construction of the calcium carbonate skeletons that form the reef's framework. In return, the algae receive a safe haven, a steady supply of nutrients derived from the coral's waste products, and access to the sunlight they crave. The acquisition of these algal partners is a delicate process, often occurring during the coral's larval stage. Free-swimming coral larvae encounter zooxanthellae in the water column and engulf them through phagocytosis. Once inside the coral cells, the algae establish themselves within specialized compartments called symbiosomes, where the intricate exchange of nutrients takes place. This exchange is a finely tuned process, regulated by both the coral and the algae, ensuring a balanced flow of resources that benefits both partners. The morphology of corals has evolved to maximize light capture for their algal symbionts. Many corals exhibit flattened, branching structures that increase their surface area, exposing more zooxanthellae to sunlight. Some corals, like those in the genus Fungia, even have translucent tissues that allow light to penetrate deeper into the coral, reaching algae located further within the colony. Others, like the mushroom coral Discosoma, harbor red fluorescing proteins that absorb blue light, which penetrates deeper into the water column, and re-emit it as longer wavelengths that the zooxanthellae can more efficiently use for photosynthesis. This delicate balance, however, is increasingly threatened by environmental stressors. Rising ocean temperatures, caused by climate change, can disrupt the symbiotic relationship between corals and algae, leading to coral bleaching. During bleaching events, stressed corals expel their zooxanthellae, losing their vibrant colors and their primary source of energy. If the stress persists, the corals can starve and die, leaving behind a ghostly white skeleton. Ocean acidification, another consequence of rising carbon dioxide levels, further exacerbates the problem by hindering the coral's ability to build its calcium carbonate skeleton. Giant clams, the titans of the bivalve world, also rely on a symbiotic dance with algae for their survival. These massive mollusks, found in the warm, shallow waters of the Indo-Pacific, can grow to over 4 feet long and weigh over 500 pounds. Their colorful mantles, exposed to the sunlight filtering through the clear waters, harbor a diverse community of symbiotic algae, primarily dinoflagellates of the genus Symbiodinium, similar to those found in corals. The clam's anatomy is perfectly suited for this symbiotic partnership. The mantle, the fleshy tissue that lines the clam's shell, is not only brightly colored but also highly folded, increasing its surface area and maximizing light capture for the algae. The mantle also contains specialized cells called iridocytes, which reflect and scatter light, further enhancing the algae's access to sunlight. The algae, in turn, provide the clam with a significant portion of its nutritional needs. Through photosynthesis, they produce sugars and other organic compounds that are transported to the clam's tissues, supplementing its filter-feeding diet. This extra energy boost contributes to the clam's impressive growth and longevity, with some species living for over a century. The giant clam Tridacna gigas, the largest living bivalve, is a prime example of this symbiotic relationship. Its massive shell, often adorned with vibrant colors and intricate patterns, provides a spacious home for its algal partners. Other species, like the fluted giant clam Tridacna squamosa, have even more elaborate mantle structures, with ruffled edges and finger-like projections that further increase surface area and light capture. These examples, however, are just a glimpse into the diverse world of symbiotic photosynthesis. Sponges, the simplest multicellular animals, often harbor a variety of photosynthetic symbionts, including cyanobacteria, green algae, and dinoflagellates. These algae provide the sponges with food and oxygen, while the sponges offer protection and access to sunlight. Some sponges, like the Caribbean fire sponge Tadania ignis, even derive their bright orange color from their algal partners. Sea squirts, or ascidians, a group of marine invertebrates, also participate in this symbiotic dance. Some species, like Didymna mall, host the cyanobacterium prochlorin within their tissues. The cyanobacteria provide the sea squirts with food and oxygen, while the sea squirts offer a protected environment and access to nutrients. These diverse examples highlight the remarkable adaptability of life and the myriad ways in which animals have evolved to harness the power of the sun. From the intricate coral reefs to the humble sponges, the symbiotic dance between animals and algae plays a vital role in shaping the underwater world and maintaining the delicate balance of life in aquatic ecosystems. This intricate web of relationships, driven by the energy of the sun, underscores the interconnectedness of life on Earth and the importance of preserving these delicate partnerships for generations to come. Chapter 3. Kleptoplasty, a solar heist in the animal kingdom. The natural world is full of surprises, constantly challenging our preconceived notions about the boundaries between different forms of life. One of the most fascinating examples of this blurring of lines occurs in the ocean, where certain animals have evolved a remarkable ability to harness the power of the sun, not through their own cells, but by stealing the photosynthetic machinery of algae. This intriguing process, known as kleptoplasty, is a unique form of symbiosis where animals acquire and utilize chloroplasts, the photosynthetic organelles of algae, to supplement their energy needs. Unlike other symbiotic relationships, where both partners coexist and contribute to each other's survival, kleptoplasty involves a more direct form of acquisition, where the animal effectively hijacks the algae's photosynthetic capabilities. Elysia chlorotica, the eastern emerald Elysia sea slug, is a prime example of a kleptoplastic animal. This vibrant green sea slug, resembling a small, leaf-shaped creature, has captivated scientists for decades with its ability to become photosynthetic. Its kleptoplastic prowess begins with its diet. Elysia chlorotica feeds on the yellow-green alga Vaucheria litorea, piercing its cell walls with a specialized feeding structure called a radula and sucking out the cell contents. However, instead of fully digesting the algae, Elysia chlorotica's digestive system has evolved a unique adaptation: specialized cells lining its extensive digestive diverticula. These cells can selectively engulf whole chloroplasts through phagocytosis, a process where the cell membrane extends and surrounds the chloroplast, forming a protective vacuole. These vacuoles then transport the chloroplasts to the sea slug's own cells, primarily those in the digestive tract's peripodia, where they become integrated into the animal's tissue. But the theft of chloroplasts is only the first step. Maintaining these stolen organelles and keeping them functional within an animal cell is a far greater challenge. Chloroplasts are complex structures, requiring a constant supply of proteins encoded by genes within the alga's nucleus. Remarkably, Elysia chlorotica has overcome this obstacle, at least partially, by acquiring some of the algal genes through horizontal gene transfer. This process, rare in multicellular organisms, involves the integration of foreign DNA into the animal's own genome. While the exact mechanisms of this transfer are still under investigation, it appears the sea slug can incorporate and express algal genes, producing proteins essential for chloroplast maintenance and function. This borrowed genetic machinery allows the chloroplasts to remain active within the sea slug's cells, photosynthesizing and producing sugars for extended periods, sometimes up to 9 months. This prolonged functionality of stolen chloroplasts distinguishes Elysia chlorotica from other sacoglossan sea slugs, which exhibit shorter periods of kleptoplasty. Elysia chlorotica's physical characteristics also play a crucial role in its kleptoplastic lifestyle. Its flattened, leaf-like body, reaching up to 3 inches in length, maximizes its surface area for light absorption. Its translucent skin allows sunlight to penetrate its tissues, reaching the embedded chloroplasts. The slug's black beady eyes, located close together on its white head, along with its rhinophores, chemosensory organs that look like ears or antennae, help it navigate and find its algal food source. The green serrata, leaf-like appendages covering its body, contain branches of the digestive gland and house the stolen chloroplasts. These adaptations, combined with its kleptoplastic abilities, allow Elysia chlorotica to thrive in its salt marsh habitat, potentially going without food for extended periods, thanks to the energy provided by the stolen sunlight. These remarkable sea slugs, which live for about a year, are hermaphrodites, possessing both male and female reproductive organs. They must mate with another individual to produce an egg mass, which is laid in tiny spiral coils on the algae they inhabit. While Elysia chlorotica is perhaps the most well-known example of kleptoplasty, it's not alone in this remarkable adaptation. Other sacoglossan sea slugs, such as Costiciella curashime, affectionately known as the leaf sheep, Elysia crispata, Placabranchus ocellatus, Costiciella ocellifera, Theridia gracilis, Costiciella curashime, Alderia modesta, Lobager voridis, and Oxino antiarum, also exhibit varying degrees of kleptoplasty. These sea slugs display a range of chloroplast retention times, from short-term retention, where the chloroplasts remain functional for a few days or weeks, to long-term retention, as seen in Elysia chlorotica. The variation in the duration of functional kleptoplasty among these species is an area of active research, as scientists strive to understand the evolutionary drivers and ecological implications of this adaptation. One challenge in characterizing the duration of photosynthesis in these sacoglossans is the difficulty of replicating natural conditions in the lab and accurately measuring photosynthetic activity over extended periods. Phylogenetic studies, which examine the evolutionary relationships between different species, have played a crucial role in unraveling the evolutionary history and distribution of kleptoplasty in sacoglossans. These studies have revealed that long-term plastid retention, as seen in Elysia chlorotica and a few other species, evolved independently multiple times, suggesting a strong selective advantage for this adaptation under certain environmental conditions. The morphology of these sacoglossans, particularly the structure of their digestive systems and the presence of finely branching diverticula, is thought to contribute to long-term plastid retention by providing a high surface area to volume ratio for accommodating the chloroplasts and facilitating the transfer of photosynthates and other nutrients. Symbiosis roscafensis is another example of a symbiotic relationship involving photosynthesis. Kleptoplasty and other forms of symbiotic photosynthesis have significant ecological implications. These adaptations influence the food web dynamics by creating new pathways for energy flow. Kleptoplastic animals, by partially or fully relying on sunlight for energy, reduce their dependence on traditional food sources, potentially altering predator-prey relationships and competition for resources. Environmental factors, such as light availability and nutrient levels, play a crucial role in the success of kleptoplastic animals. In environments where light is abundant and food sources are scarce, kleptoplasty offers a distinct advantage, allowing these animals to thrive where others might struggle. The study of kleptoplasty and related adaptations continues to reveal surprising insights into the adaptability of life. Ongoing research is exploring the genetic mechanisms of horizontal gene transfer, the intricacies of chloroplast maintenance within animal cells, and the ecological consequences of this unique form of symbiosis. These investigations not only challenge traditional biological classifications but also hold potential for future applications in biotechnology and medicine. The ability to engineer photosynthetic capabilities into animal cells, for example, could have implications for developing new sources of renewable energy or creating self-sustaining tissues for medical applications. The sea slug's solar heist, a testament to the ingenuity of evolution, offers a glimpse into the incredible diversity of life and the surprising ways in which organisms can adapt to their environment. Chapter 4. Nature's Solar Innovators, Beyond Kleptoplasty and Symbiosis. The emerald green sea slug and the symbiotic corals, with their remarkable adaptations for harnessing solar energy, represent just the tip of the iceberg in the fascinating world of photosynthetic animals. Beyond kleptoplasty and symbiosis, nature has devised even more ingenious strategies for capturing and utilizing the sun's power, showcasing the remarkable diversity and adaptability of life on Earth. The oriental hornet, Vespa orientalis, a formidable insect found across parts of southern Europe, northeast Africa, and Asia, demonstrates a unique approach to solar power generation. Unlike plants and algae, which use chlorophyll to capture light energy, the oriental hornet employs a specialized pigment called xanthopterin. This yellow pigment, found in the hornet's cuticle, the hard, outer layer of its exoskeleton, plays a crucial role in converting sunlight into electricity. The hornet's exoskeleton isn't just a protective armor, it's also a sophisticated light-trapping device. Microscopic grooves on the surface of the cuticle act like miniature solar panels, channeling sunlight towards the xanthopterin-rich layer beneath. As photons, the fundamental particles of light, strike the xanthopterin molecules, they excite electrons, generating a flow of electrical current. This bio-generated electricity isn't just a curious side effect. It appears to be linked to the hornet's unique activity patterns. Unlike most wasps, which are most active in the cool morning hours, the oriental hornet buzzes with activity during the hottest part of the day, coinciding with peak sunlight intensity. This suggests a direct correlation between the hornet's solar-powered abilities and its metabolic demands. While the exact mechanisms and functions of this bioelectricity are still under investigation, researchers believe it may play a role in various physiological processes, such as larval development, nest thermoregulation, and even flight. Some studies have even suggested that the xanthopterin pigment may act as a light-driven proton pump, generating a proton gradient across the cuticle that could be used to power ATP synthesis, the fundamental energy currency of cells. The pea aphid, Acyrthosiphon pisum, a tiny but ubiquitous agricultural pest, takes a different approach to solar energy utilization. These small insects, found on a variety of plants, harbor a surprising secret. They can synthesize carotenoids, pigments typically found in plants and algae, but rarely in animals. These carotenoids, which give the aphids their characteristic colors ranging from white to orange to green, appear to play a role in light energy absorption and potential conversion into ATP. Studies have revealed a correlation between aphid color and ATP levels. Green aphids, with the highest concentration of carotenoids, exhibit significantly higher ATP levels than white aphids, which lack these pigments. Orange aphids, with intermediate carotenoid levels, show an intriguing pattern. Their ATP production increases in the presence of light, suggesting a potential link between carotenoid-mediated light absorption and energy production. While the precise mechanisms of this energy conversion are still under investigation, the presence of carotenoids in these aphids represents a unique adaptation in the animal kingdom, blurring the lines between plant and animal energetics. Researchers are exploring the possibility that these carotenoids may act as accessory pigments, transferring absorbed light energy to other molecules involved in ATP synthesis, or even directly participating in the electron transport chain, the core process of energy production in cells. These diverse examples, ranging from the buzzing oriental hornet to the delicate pea aphid, demonstrate the remarkable range of photosynthetic adaptations in the animal kingdom. They highlight the ingenuity of nature and the diverse ways in which life has evolved to capture and utilize the energy of the sun, blurring the lines between plant and animal, and challenging our traditional understanding of biological classifications. These adaptations, honed over millions of years of evolution, have profound ecological implications, influencing food web dynamics, shaping ecosystems, and highlighting the interconnectedness of life on Earth. The evolutionary history and significance of these adaptations are still being unraveled. Scientists are investigating the genetic mechanisms underlying these unique traits, exploring the ecological factors that drive their evolution, and evaluating their potential for future applications. The ability to engineer photosynthetic capabilities into animal cells, for example, could have far-reaching implications for developing new sources of renewable energy, creating self-sustaining tissues for medical applications, and even enhancing agricultural productivity. The study of photosynthetic animals, with their remarkable ability to borrow the sun's power, continues to inspire new avenues of research and promises to unlock further secrets of the natural world. This field, still in its early stages of exploration, holds immense potential for future discoveries, offering a glimpse into the incredible diversity and adaptability of life, and paving the way for innovative solutions to some of humanity's most pressing challenges. Chapter 5. The Future of Food and Medicine. Can we harness the power of photosynthesis in animals? The discovery that some animals can harness the power of the sun, much like plants, has sparked a wave of excitement in the scientific community. This isn't just a fascinating biological quirk, it's a potential game-changer for the future of food and medicine. Imagine a world where we could grow meat in a lab, not by feeding cells traditional nutrients, but by simply exposing them to light. Or picture doctors treating diseases by delivering targeted doses of oxygen to damaged tissues, again, using just light. These scenarios, once confined to science fiction, are now within the realm of possibility, thanks to our growing understanding of photosynthetic processes in animals. Let's start with the future of food. Lab-grown meat, also known as cultured meat or cellular agriculture, offers a promising alternative to traditional animal agriculture, with the potential to reduce our environmental footprint and improve animal welfare. However, scaling up lab-grown meat production faces a significant hurdle: oxygen delivery. Growing thick tissues in a lab requires a constant and efficient supply of oxygen to every cell. Current methods struggle to achieve this, limiting the size and complexity of the meat that can be grown. Photosynthesis offers a potential solution. Imagine incorporating photosynthetic cells, perhaps derived from algae or even engineered animal cells, into the lab-grown meat. These cells, when exposed to light, could generate their own oxygen, providing a localized and sustainable supply within the growing tissue. This could overcome the oxygen delivery limitations, allowing for the production of larger, more complex cuts of meat. Furthermore, these photosynthetic cells could also consume carbon dioxide, a waste product of cellular respiration, further enhancing the sustainability of the process. This vision could be realized through the use of bioreactors, specialized vessels designed for cell culture. These bioreactors could be equipped with optimized light delivery systems, ensuring that every photosynthetic cell receives the light it needs to function efficiently. The light source could be tailored to the specific needs of the photosynthetic cells, using specific wavelengths and intensities to maximize oxygen production and minimize any potential damage to the animal cells. But the potential of photosynthetic animal cells extends far beyond the dinner table. In medicine, the ability to deliver oxygen to specific tissues on demand could revolutionize the treatment of various diseases. Take heart disease, for example. Oxygen deprivation, or ischemia, is a major factor in heart attacks and other cardiac conditions. If we could introduce photosynthetic cells into the heart tissue and expose them to light, we might be able to stimulate localized oxygen production, potentially mitigating the damage caused by ischemia and promoting tissue repair. This concept of targeted oxygen delivery could also have profound implications for regenerative medicine. Growing new tissues and organs in a lab, a key goal of regenerative medicine, requires a precise and controlled environment. Photosynthetic cells could play a crucial role in this process, providing a localized and on-demand oxygen supply to the developing tissues, enhancing their growth and viability. However, realizing the full potential of photosynthetic animal cells in medicine faces several challenges. Introducing and maintaining chloroplasts, the photosynthetic organelles, within animal cells long-term is no easy feat. Animal cells typically recognize foreign organelles, like chloroplasts, and destroy them. Furthermore, even if the chloroplasts are successfully introduced, they may not function optimally in the warmer environment of animal cells, which are typically cultured at higher temperatures than plant cells. Ongoing research is tackling these challenges head-on. Scientists are exploring various strategies to enhance the compatibility between animal cells and chloroplasts. One approach involves genetic engineering, modifying the animal cells to express proteins that protect the chloroplasts from degradation and provide the necessary support for their function. Another avenue of research focuses on identifying and utilizing heat-tolerant chloroplasts, such as those found in algae that thrive in hot springs. These chloroplasts, adapted to higher temperatures, could potentially function more efficiently within animal cells. Researchers are also looking to nature for inspiration, mimicking natural processes like horizontal gene transfer or kleptoplasty to achieve stable integration of photosynthetic capabilities into animal cells. Horizontal gene transfer, the process by which genes are transferred between unrelated organisms, could be used to introduce algal genes essential for chloroplast function into animal cells. Kleptoplasty, the process observed in sea slugs, where chloroplasts are stolen from algae and incorporated into animal cells, could provide valuable insights into how to maintain these organelles within a foreign environment. The recent breakthrough by researchers at the University of Tokyo, where they successfully introduced photosynthetically active chloroplasts from algae into hamster cells, is a significant step forward. While the chloroplasts only functioned for a few days, this achievement demonstrates the feasibility of this approach and opens up new possibilities for future research. While the potential benefits of photosynthetic animal cells are enormous, it's crucial to consider the ethical implications of these bioengineering advancements. Introducing foreign organelles or genes into animal cells raises questions about the potential for unintended consequences, both for the animals themselves and for the environment. Careful consideration of these ethical issues, alongside rigorous scientific research, is essential to ensure the responsible development and application of these technologies. The ability to harness photosynthesis in animals is a tantalizing prospect, with the potential to revolutionize food production and medicine. While significant challenges remain, ongoing research is paving the way for a future where we can tap into the power of the sun to improve human health, address global food security challenges, and create a more sustainable world. This journey of discovery, inspired by nature's ingenuity, promises to unlock new frontiers in biology and bioengineering, pushing the boundaries of what's possible and offering a glimpse into a future where the lines between plant and animal become increasingly blurred. Several organisms have evolved astonishing ways to harness sunlight, from symbiotic partnerships with algae to the outright theft of chloroplasts. This fascinating intersection of plant and animal biology reveals nature's ingenuity and adaptability. These discoveries have far-reaching implications, potentially revolutionizing fields like lab-grown meat production and targeted oxygen delivery in medicine. Imagine self-sustaining tissues powered by light. The future of this research is bright, offering innovative solutions to global challenges. What are your thoughts on photosynthetic animals? Share your questions and ideas in the comments below. Like, subscribe, and suggest topics for future explorations. Keep asking questions. The wonders of the natural world are waiting to be discovered.